this morning? That's okay. All right, so we're going to go through the integumentary system in a little bit more detail. I just kind of gave you an introduction last time. And we'll try to cover everything but the mammary gland and the hoof. Both of those are a bit more challenging. And we'll save those because I think you are, excuse me, I think you're a little distracted right now. So we'll try to keep it simple. So what I'm going to do is what I've been doing, and I hope it has helped this year, is I'm just going to go through the handy dandy lecture notes. And that way, everything that I cover is in order of the notes, and you'll have a nice reference. I've highlighted things that I wanted to, bring to your attention. So we'll start with this cutis and subcutis. And why you need to know that is because, well, you give sub-Q fluids, all right? And do you know what that means? You know what? Well, that's a shortened version of subcutis, sub-Q, and it's the hypodermis. So if that is the hypodermis, that means it's under the dermis. So remember what you skinned down in gross anatomy was the epidermis, because the hair went off with it, and then the underlying dermis, that dense irregular connective tissue. So all of that came off together in a big sheet, and where your blade was separating that skin, that cutis, from the underlying deep fascia, which we've been looking at in anatomy lab this week, those big sheets of deep fascia. In between those two layers is the subcutis, and that's where you get all that fat deposit. Some of you had dogs with more fat deposit than others, and where you have your cutaneous muscles. And that's where you can pick up the skin, you can pick up the cutis on your pets, and you can lift that up, and that's that space where you put your needle in to deliver your sub-Q fluids. So now you know what sub-Q means. Okay, we've looked at these strata before in the epidermis, and we'll just look at them in slightly more slightly more detail. So the stratum basale, we already mentioned that it's a single layer. It's columnar to cuboidal cells. So some of these are very much columnar, and sometimes they're more cuboidal. It's a single layer of stem cells, and because of obliquing, it at times will look more than a single layer. It will look, let's see if I can find an oblique area. It'll look several layers thick, like right here. Okay, because you can see all the little melanocytes in here. And so this looks like a stratified stratum basale, but that's just obliquing. Okay, so it's really just a single layer thick, like what you can see right in this area right here. That's a little blurry right there. Okay, so single layer of stem cells. Also, that's where your melanocytes like to hang out. And remember, melanocytes are non-keratinocytes, meaning they aren't part of that stratified squamous epithelium. And you can pick these guys out because they have melanin associated with their cytoplasm and all their little cytoplasmic processes that go out. Other non-keratinocytes would be your tactile epithelioid cells and your Merkel cells. And I've got a little schematic where I can show you that in just a second. So we have our stratum basale, we've got the tactile epithelioid cells, the melanocytes that also hang out in that stratum basale. Then next, as these basal cells, these stem cells divide, they'll keep a stem cell down here, and then they'll send one of the daughter cells up here, and those will become the cells of the stratum spinosum. That's usually the thickest layer in skin, not always, but usually. And it's called the prickle cell layer, or the spinous cell layer. And that's because we get shrink artifact between the cells in these INTER, intercellular spaces. And the shrink artifact exposes the little linker proteins that span that intercellular space between the desmosomes. And the desmosomes are connected to that intracytoplasmic cytoskeleton inside those cells. Sometimes you can even see those spinous processes down here between these cells of the stratum basale, because they also have desmosomes between themselves, and then they have hemidesmosomes where they attach to that basement membrane. The stratum granulosum, you don't always see. It's present in soft keratin, but you don't always see it. What the heck is soft keratin? Well, soft keratin is skin that sheds or desquamates. Okay. Hard keratin is an epidermal product that doesn't shed individual squames. So hair, hoof, and horn would all belong in the category of hard keratin, but soft keratin would be, you know, just everything else like on your integument that just sheds little tiny little squames, little individual keratinocytes. You know, that's what creates all that dust in our household. I was reading something interesting yesterday. You know, the integumentary system is something that you can become a board certified dermatologist. You can specialize in it. It's something that's really important. Clients tend to bring their animals to you when something's wrong with the integumentary system on their animal. So that's good. So it's a moneymaker. And one of the things I read is that there are enzymes up here in this stratum granulosum that contributes to the breakdown, enzymatic digestion or breakdown of those desmosomes in between the individual keratinocytes so that we can slough individual squames. So when that process breaks down, say like if your skin becomes too dry, that's when you can get, you know, like plaques of skin, you know, peeling versus just the individual little tiny squames like this sloughing off. So I thought that was pretty interesting. Now this stratum granulosum has these keratohyalin granules. Those are intracytoplasmic. Those are going to combine with the keratin filaments inside the cell and create that brick and mortar, more like rebar. The filaments, the keratin filaments are more like rebar, and the keratohyalin granules are more like your mortar, and that goes together to make, you know, a concrete reinforced with you know, iron rods with rebar in it. These cells will also secrete lamellar granules, which we can't see in the intracellular spaces, and also those enzymes that will help break down those desmosomes. So that's kind of a lipid or waterproofing effect, both from the keratinocytes sort of turning into you know, concrete and rebar, and then that you know, that oily lipid type substance from those lamellar granules will help with that sealing off, getting that waterproofing. I guess that's kind of like waterproofing your deck, or, you know, putting a sealant on your outside of your brick house. It just helps prevent water penetration. So it's a pretty cool system. Now the stratum lucidum is next. You won't always see it. It's inconstant, and it's fairly rare, and we don't have an example of that, but it's just a clear layer that would be between the stratum granulosum and the stratum corneum, and sometimes it's called the clear layer, and it's pretty cool when you see it because you're like, oh wow, I've heard about that and didn't believe it, but there it is. Next is the stratum corneum. You should be fairly familiar with that. Sometimes it's called, you know, you'll have like the stratum corneum right here, this acidophilic layer of dead cells that have gotten rid of their organelles and nuclei. But then you'll have all of this that's kind of, you know, lifting off, kind of wanting to peel off right here. And an extra term, because, you know, you can never have too many terms in histology. So an extra term would be for that lifting off, starting to slough layer, the stratum disjunctum. It's become disjoined from the rest of the underlying stratum corneum. So this would all be kind of stratum disjunctum right here, where it's lifting off. And look at that, you can see the little individual squames outlined there, and you can even see the residual melanin pigment that's inside those squames. Okay, so that's all your strata in more detail. We already looked at the non-keratinocytes on the last lab. We chased around on some of them. So they're hard to find. Melanocytes are the easier ones to find because you've got that pigment to help you, you know, visualize them, to, you know, kind of zoom in on those guys. The tactile epithelial cells, they're kind of difficult, I think mostly because of obliquing. They tend to have kind of a round nucleus and a spherical cytoplasm, and they should be on that basement membrane where they can be in contact with underlying nerve axons. And then there's the dendritic cell, or the Langerhans cell, or the epithelial macrophage, and they move around, and they can kind of be in any layer. So when you see shrink artifact in the different layers, like stratum spinosum, you might want to think, well, that could be a tactile epithelial cell there. excuse me, a Langerhans cell there, or a macrophage. And then the tactile epithelial cells, again, hopefully they would be along the basement membrane. I like the shape of this one, but when I zoom in on it, it's got a little melanin associated with it, which makes me a little nervous that maybe I'm just looking at a bit of a melanocyte, but that kind of gives you the idea of what it would look like. Okay, let's see. Melanocytes have the melanosomes inside of them, and they secrete by the cytokine mode of secretion. So you need to know that. You need to be familiar with the melanosomes being the prepackaged melanin pigment that they will pass off via cytokine secretion to the surrounding cells of the stratum spinosum, and then those guys will transport that melanin pigment up on the outer surface of their nucleus in the direction that the UV rays would be penetrating, so they protect their chromosomes, which I think is one of the coolest things you can see in the body. Here is a melanocyte here. We can't really see its cytoplasmic processes, but you can see he's been pretty busy passing off melanin packets, and here's a little bit of a melanocyte here. All right, so I think that gets us through the non-keratinocytes and the things we needed to say about that. What do we need to look at in the dermis? Or another name for the dermis is corium. And that's important because we'll switch to that term corium when we talk about the hoof. Okay, so keep that in mind. Dermis is the same thing as corium, especially when you're talking about the hoof. Now, the dermis has a couple of layers that we want you to be familiar with. They're not really outstanding in this slide, but they're a little bit easier to understand here. This is the dental pad of the ruminant. And even at this power magnification, I can see that I've got a lot of dense irregular connective tissue here, and it looks less dense. It's starting to look loose right here, okay, in what we were calling the dermal papilla. So where you have just outside of the basement membrane, if you have loose irregular connective tissue, and you'll always have that, but it may be so thin that it doesn't really stand out like in that other slide, you'll have the papillary layer. And then outside of that is called the reticular layer. So those are the two subsets of the dermis. So this would come off when you're skinning that epidermis and dermis off of the cadaver. Now, this loose irregular connective tissue is heavily vascularized, and remember, it makes those interdigitations with the epidermal pegs, and you've got your dermal papillae. Again, like if you interdigitated your fingers, so here's one hand, here's a finger of your one, say like my right hand, so here's a finger, a finger, a finger, a finger, and then my left hand here would be like a finger and a finger and a finger and a finger. So I'm just interdigitating those two. That's what we're doing. We're interdigitating to get these dermal papillae that are heavily invested with capillary beds. Got to get that all the way up here so that we can have simple diffusion of gases and nutrients to support this living stratum spinosum. Okay, hypodermis you'll see in gross anatomy. You have seen it, and that's also called the subcutis. Now, moving on to skin appendages, we've got the hairs to look at. So I'm going to switch over here to the palpebra. This is the palpebra of the eye. And I'm going to take a breath and a swig of hot tea. Okay, ooh, that's good.
While Dr. J is taking a swig of her tea, I just wanted to mention to the students, you know, why do we learn about these layers of the epidermis? And it clinically will be important. For example, I'm sure many of you have heard of diseases like pemphigus vulgaris. Pemphigus is basically where our body attacks those desmosomes that Dr. Jarrett was talking about. So clinically, to educate your clients, if you have a skin disease, there are times that you will actually, use the histology to teach those clients that what holds those cells together. Those cadherins actually get immunologically targeted from our body and cause those blisters that we see in those diseases.
Yeah, it's really cool. And they'll talk to you about that in great detail in dermatology. Yeah, it's really neat. I just saw some students not that long ago talking about a bacterial folliculitis. So let's look at some hair follicles, because you're going to be looking at bacterial folliculitis, or you'll even be looking at at parasites that live in those follicles, demodectic mites. All right, so skin appendages. Well, we have hairs, and hairs have three subdivisions: the shaft, the root, and the hair bulb. The shaft is what protrudes off of the surface of the epidermis. The root is within the follicle, which is an invagination down into the dermis. And then there's the hair bulb, and that's how that whole hair follicle attaches to and kind of hangs on to that dermal papilla. So there's that papilla, that papillation, that nipple-like protrusion. That's a concept I really want you to think about and cement in your head, because I can't explain the hoof to you if you don't really understand the dermal papilla concept of how this configuration of the interaction of the epidermis with a dermal papilla, this kind of little conical configuration, gives rise to the hair and the hair follicle. And that same theme is going to come back in the hoof. So stay tuned for that. Now, what the hair shaft also has layers. There's actually more layers in this than we had to learn, you know, back in the old school days, back in the 80s. But we've condensed it, so we're going to look at the cuticle, the cortex, and the medulla of the actual hair shaft. Then we're going to look at the subcomponents of the hair follicle: the internal epithelial root sheath, external epithelial root sheath, glassy membrane, dermal root sheath, dermal papilla. And then we'll look at the germinal hair matrix cells, and the dividing cells that give rise to that hair. So let's take a look at all of that using this slide right here. I'm going to move these out of the way temporarily so I can have more surface area here. Okay, so this is a hair that is protruding off of the surface of the skin. So what do we call that? And that is the shaft, right? Okay, so we said the part that was down in the hair follicle is the root. So here's a part down in the hair follicle. So that's root, shaft, and then hair bulb. So here's a hair bulb here, and you're going to get all kinds of different configurations of hair bulbs due to sectioning artifact. So here's a hair bulb, a little bit right here. This is a more classic hair bulb here. Here's a classic hair bulb. There's a bulb everywhere, hair bulb. Okay, we'll be zooming in on those in just a second. Now the hair shaft has the three layers: the cuticle and the cortex and the medulla. So here is, this cuticle isn't showing up all that well, but you can kind of see a shingle-like arrangement. There's one that's a little better to look at in just a second. Here's that dense cortex, and then here is that more air-filled, not as densely packed keratinocytes. They make up the medulla. So yes, all the components of the hair, the cuticle, the cortex, the medulla, those are all keratinocytes, because this is something that's made from the epidermis. Okay, so over here, this one, you can see that cuticle better. It actually will come into view. So see how it looks like the shingles on a roof? And notice that the little edges of the overlapping shingles are pointing upwards. Here you can see that little edge is pointing upwards towards the skin surface. Okay, and there's a reason for that. And we'll look at the reason why in just a minute, why those would point upwards. Okay, so then if that's the layers of the hair shaft, then what are the layers of the hair follicle? Well, let's zoom in down here. Let's see, we can look here. Now remember, the follicle is just the invagination of that epidermis down into the dermis. So if I took my finger and just push this epidermis down in here, I would still have all those layers of the epidermis. Okay, so what are those layers? Well, here would be our stratum corneum. This is the hair right here. This would be our stratum corneum of the hair follicle. This would be our stratum spinosum, and then our stratum basale of the hair follicle, and then our basement membrane would be out here. But so those are just standard layers of the stratified squamous epithelium, but we change the name when it becomes associated with a hair follicle. So we're going to call this not the stratum corneum. We're going to call this the internal epithelial root sheath. And then we're not going to call this the stratum spinosum and the stratum basale. And boy, you can really see that division right there between the spinosum and the basale. We're going to call that the external epithelial root sheath. And that's really easy because this is epithelium and this is internal and that's external. And hey, we can do that. And that's associated with the hair. So this is internal or closer to the hair, and this is external or further away from the hair. Then we have what was next, the glassy membrane. That's just that funky name for a thickened, more elaborate basement membrane. We heard that term in the ovary. So we're going to use it again. We're going to recycle that term here in the hair follicle. Then the dermal root sheath is just that surrounding dermis, kind of like the thecal layers around a follicle. It's just that around a follicle. It's just that specialized little bit of dermis just outside of the glassy membrane or that specialized basement membrane. And then there's the hair matrix. Now that's where all the excitement happens, the hair matrix. And that's around that bulb of the follicle. And the hair matrix is really, it's all about all of the layers of the epithelium over that bulb. Now let's see if I can find, I know I had some bulbs that were more classic. And this one, we'll use this one. So the bulb, it's like an upside down funnel. So this is epidermis, then this is dermis right here. And you can see how this upside down funnel, here's the little rim of the funnel right there. Then here's the basin of the funnel. And then here's the opposite rim of the funnel. Now remember, it's a 360-degree structure. So that funnel is cupping a little dermal papilla. The funnel is the epidermis, and that is specialized to form that upside down funnel that cups around or hangs on to that papilla, that dermal papilla. And this helps hold that hair in place, okay? And these layers are going to give rise to different portions of the hair. So your germinal matrix cells are really just your stratum basale, your stem cells that form that ledge of the funnel, okay, in a 360-degree configuration. So the stem cells kind of in the middle of that upside down funnel configuration, they'll give rise to the medulla of the hair. A little further out, they'll give rise to the cortex of the hair, and a little further out, the cuticle of the hair, and then further out, the will be continuous with those inner and outer root sheaths. And I'm looking to see if I have any other, yeah, here we go. This one shows it. We just don't see that where that dermal papilla goes up inside that upside down funnel. That's just obliquing artifact. But here are all our stratum basale stem cells. And you can see this little peak right here. This area is going to give rise to hair medulla. Then this area here and here will give rise to hair cortex. And then out here will be continuous with the inner layer of the inner epithelial root sheath. And out here will be continuous with the external epithelial root sheath. So it's a way cool way to make a hair. And we need to understand that because it's also how we make the structures of the hoof wall. And those are incredibly strong structures. And if you think about it, a horse is running around on its middle finger. So it's a really way cool way to make up that hoof wall so that it can support all that weight. All right, so that's how we make the hair. Now the pigment of the hair will come, remember the melanocytes reside in that stratum basale. So that's why the hair matrix cells, which is also called the hair germinal epithelium down here, it'll often stain pretty dark or pretty have a lot of melanin in it because that's where the pigment comes from. Those melanocytes that are in that stratum basale of the hair bulb. Now to practice looking at at hairs or at hair follicles, I like to find hair follicles where the hair is missing, has maybe fallen out during sectioning. So there's a hair in here, that's just through the cortex, so it's oblique, but there's not a hair in this follicle. Now this would be your internal root sheath right here. This would be your external root sheath. Your glassy membrane would be out here, and then your dermal root sheath would be out here. The little cuticle is right there. So the internal epithelial root sheath also has a cuticle. Let's see, I think we can see it. There was one of them where it showed up nicely, and I wanted to share that with you. Where'd he go? Where'd he go? Here, this one. So the inner layer of that internal epithelial root sheath, it's not really the one. It's also got a cuticle, and it's hinged downwards. Its little shingles are hinged downwards, and the shingles on the hair cortex are hinged upwards. And you can see how if you tugged on a hair, then, I wonder, where did that pretty one go? Then that would hold that hair in place, help hold that hair in place, because those two shingles would oppose each other beautifully. I thought I had a place to show you. Dadgummit, I can't find it. Okay, well, it's not an important concept. It's just a way cool one to look at when you can see those shingles going in opposite directions. All right, what do we have left? We talked about pigmentation. And let's see, erector pili muscle. Erector pili muscle don't really show up very well on this slide. And I was thinking about that. And I think it's because the erector pili muscle kind of make the hairs stand up under sympathetic tone, this smooth muscle, and it makes the hair stand up, especially when you're cold or even afraid. And this is an eyelid, and I think that just doesn't happen very much on the eyelid, which is, our hairs don't really stand up when we're scared on the eyelid. So I don't think we're really going to see those erector pili muscles. I think we might see them in another slide, and I'll point those out later. Okay. There's things here in the notes that I'm not going to go over, like hair growth or the hair cycle, anagen, catagen, and telogen, but those are underlined terms, so you might want to know those. Primary follicles, secondary follicles, single follicles, and compound follicles, not really going to go over those, but we will look at individual hairs and practice them for just a minute or two, and then we'll move on. So we looked at those root sheaths. Let's look, see if we can find some hair. Here we go. So this hair is obliqued. Okay, so we're only seeing cortex. And how I know that is because here's cortex here. Okay, and you can see those opposing layers of shingles right there. So they're going in this way on the cuticle of the hair, and they're going that way in the cuticle of the hair follicle. So they oppose one another. Helps keep that hair in there. This is that lighter, more acidophilic staining, kind of airy, loosely packed keratinocytes that make up the medulla. So medulla, cortex, cuticle, then here's the cuticle of the inner root sheath, inner epithelial root sheath, external epithelial root sheath, glassy membrane, and then dermal root sheath. So you just have to kind of drive around and practice, because you're going to see all these obliques like this, and that's really kind of just through the stratum corneum, I think. You won't always see everything that you want to see, so you have to drive around until you find what you want to see to practice. So this one is a good transverse section. I can see the cortex and the medulla of the hair, but this one I can really only kind of see cortex. Here I can see both. There I can only see cortex. There, gosh, I'm not real sure what that is. I think just oblique through stratum corneum. So you get the idea. You got to drive around, and there'll be a lot of things where you'll be like, I don't know what the heck this is, but someone is really trying to get me. They just called my phone and my landline. Oh my goodness. Is everything going okay? Can y'all hear me all right?
Yep.
Okay. All right.
Yep.
I don't know what's going on. I don't know who's trying to get me, but they really want to. So I wanted to make sure it wasn't you, Dr. K, trying to tell me you're talking to nobody, you went offline or something like that.
No, you're good.
Okay. Well, I just wanted to check. Let's see. One of the next thing in our notes is tactile hair follicles, and they're those are just whiskers, folks. So here is a huge follicle associated with a whisker. You know, when you look at your dogs and cats and the whiskers at the base have a big lump associated with it, well, here's the lump. And it's a really specialized follicle. Again, back in the dark ages in the 80s, we had to learn all the different components of these. Now we just kind of wave at them and go, wow, isn't that magnificent? And then we move on. So we've actually got these huge blood-filled sinuses associated with these tactile hair follicles, or that's why they're called sinus hair follicles, because of these blood sinuses. I don't remember why they have that, because again, we don't really teach this in detail anymore. There's even got its own little sebaceous glands associated with it here. Just beautiful. You can see how it's a very, very large follicle, even though it's obliqued here, and it have those big, strong whiskers coming out, which are also called sinus hairs or tactile hairs, because, you know, they use those to kind of feel their way in the dark. All right, next are sebaceous glands, and oh my goodness, sebaceous glands are everywhere in the skin. You can practice them in every section of hair, of skin. They're simple glands. They can be branching, they can be compound, but the main thing I want you to know is that they're alveolar, okay? That's the take home, is that the adenomere shape of sebaceous glands is alveolar. Now, really the only compound ones that you need to really become familiar with are these tarsal glands, these meibomian glands here, the specialized compound sebaceous glands that secrete meibum. So don't say they secrete sebum, say they secrete meibum. I know it seems like ha 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 splitting hairs, but it's what the ophthalmologists are going to want you to say. So that's why we're going to say that. They're meibomian glands, a specialized type of compound sebaceous glands, and they secrete meibum. The rest of these sebaceous glands associated with the skin are just regular sebaceous glands. Here you can see some of the branching, but why the heck am I calling them alveolar? You don't really see an empty lumen, right? Well, that's just it. They have a large lumen, so they are alveolar, but it's not empty. It's because it's filled with cells. So at the base of these alveoli, and the fundus are where the stem cells are, right along the basement membrane, and these cells are dividing, filling with lipids, and these cells are dying and will eventually be secreted whole with an intact cytoplasmic membrane. And that secretion is a lipid or oily type secretion known as sebum. So sebaceous glands are alveolar glands that may branch, they may be simple, they may be compound, and that they secrete sebum, and that is by the whole cell method, and that is called holocrine, whole cell method, holocrine. Sweat glands are called sudoriferous glands, and they vary greatly in appearance, as this says, and they can produce a watery secretion like sweat, which is actually called sudor. They can produce an oily secretion, or they can even produce kind of a mucousy secretion. So, and then they also make milk, okay? So mammary glands are just modified, specialized sweat glands. There are two types of secretory methods, the merocrine and apocrine. We won't really see the merocrine types in a slide today, because all of our slides are, the sebaceous glands are associated with haired skin. So that's the apocrine mode of secretion. Merocrine is another one of those modes of exocrine modes of secretions that you need to know about. The secretory packets that are packaged probably in the Golgi apparatus fuse with the cell membrane and then just release their product out into the interstitium or to the luminal surface. So they don't lose any of the cell cytoplasm with that merocrine. So it's an exocytosis. But with apocrine, they get these apical blebs where they pinch off packets of cell cytoplasm. And that is a different kind of exocrine secretion. Yes, did I hear you, Dr. K, or somebody? Nope.
I think it was just background.
Okay. All right. So why are we talking about that in detail? Well, because sometimes you'll see those apocrine blebs. So here are sweat glands, simple coiled tubular glands. They can have a very dilated lumen. They tend to have just a simple cuboidal epithelium lining them, but that can become columnar. So where can we see that? We can see that in this slide. This is the anal sac slide. So let's turn it here. This is just regular haired skin surface. There's a little bit of the rectum right here. And this is the anal sac right here. So here's its duct. So those invaginate, if you haven't seen them in gross anatomy yet, you need to. At the 4 o'clock and 8 o'clock positions, you'll see those ducts. Stick a probe in there and then make a cut with your scalpel, and voila, you'll be down in that anal sac, which will be filled with all kinds of lovely funk for you to find. Here we've got the anal sac lumen. It's just a cutaneous diverticulum, just an invagination of that epithelium, just kind of like the hair follicle, only very large lumen. And it's surrounded by glands. In carnivores, it's only surrounded by sweat glands, but in the cat, it's surrounded by both sudoriferous or sweat glands and sebaceous glands. So it's thought that's one reason why cats don't impact their anal sacs quite as frequently as dogs, because they've got that lipid oily product to help soften the secretion and help it be moved out during defecation or during times of fear, when they're brought to you in the clinic and they're frightened and they express their anal sacs all over you. But hey, then you don't have to do it. So what is that stinky, smelly gunk inside the anal sac? Well, it's just slough, stratum corneum, and then the secretions of the sudoriferous glands and the secretions of the sebaceous glands if you're a cat, and it ferments in here. It's just, look, it's just that fermented product. Same thing as in a skunk, although skunks add perfume, right? Right. And yeah, it's just that fermented, smelly, stinky product that you know, brings animals to you in your clinic because they're scooting their butt on the nice carpet and you need to express their anal sacs. Here, this shows you that these are called the glands of the anal sac. They are a type of sweat gland. They surround and empty into those anal sacs. And this shows you that they're just modified sweat glands or sudoriferous glands. And you can see that they'll have like a cuboidal to a columnar epithelium. If they are empty, then there's no pressure to cause inhibition of secretion. So like this one is filling up with secretory product. So we're getting pressure feedback and these cells are getting flattened out and they're slowing down production. But these guys don't have much of anything in their lumens, so they're maxing out production. So they'll be more of a columnar, tall columnar cells. Here you can see the little pinched off cytoplasmic packages. That's that apocrine secretion. Okay, those are the glands of the anal sac. And that's easy to remember because they just surround the anal sacs. Okay, we'll come back to that slide for a couple of other glands. The next on your list is the pinna of the ear. And it's all going to go faster now because we already know what sweat glands and sebaceous glands look like. So we just have a few things to touch on and we'll finish up. In the ear, you have what are called ceruminous glands. So again, they're sudoriferous glands. They are sweat glands, but they get a magic name change. So in the ear, they are called ceruminous glands, and you need to know that for clinics. And you need to know that earwax is called cerumen, and it is a combination of sebaceous gland and ceruminous or specialized sweat gland secretions. Okay, so what do those look like? This is a section of the pinna of the ear, and really it's just going to look like sebaceous glands and simple coiled tubular sweat glands, but we're going to call them ceruminous glands. Here's a little tiny hair follicle. So we have haired skin here. Now, one of the nice things about this slide is you can see the little myoepithelial cells out here on the outside of the epithelium lining the sweat glands. And those myoepithelial cells contract and squeeze out the secretory product. Okay, myoepithelial cells. Here are some really, really brilliant, beautiful cells associated with this sebaceous gland. So you can see how these are your stem cells here. They're starting to fill up with lipids. They're starting to die. Here they have died. They've got pycnotic nuclei, and they're just filled with huge lipid packets. And they're going to release their product into the hair follicles. So sebaceous glands in haired skin tend to release their product into the hair follicle. Sebaceous glands can also empty directly onto the haired surface, but if there's a hair follicle around, that's where they're going to want to release their secretory product. Eyelids, we looked at the tarsal glands or the meibomian glands. They're in that tarsal plate, that dense irregular connective tissue. We also saw just the regular sebaceous glands and sweat glands in that eyelid. Now, the ophthalmologists are going to call them the glands of Zeiss. They're going to call the apocrine sweat glands the glands of Mole, and they're going to call the eyelashes cilia. We're not going to do that to you right now. We're just delighted if right now you can recognize that tarsal gland and know that it's called a meibomian gland, that it's a great big compound sebaceous gland, and that it secretes meibum. We'll let the ophthalmologist tell you that sebaceous glands are called the glands of Zeiss and the apocrine sweat glands are called the glands of Mole. Yeah, we'll let them do that. Okay, anal sacs and circumanal glands, and I think we'll be done. Let's go back here to that anal sac slide. And circumanal glands are important because they are a frequent site of tumors in canines, both intact males and females. And when they get older, they can get adenocarcinoma of the circumanal glands. So you need to know what they are. And unfortunately, we don't entirely know what they are. I call them little Christmas stockings. You come over here and look at this right here. See how this kind of looks like a Christmas stocking hanging at the mantle? Here's a little bit of one here, just a little bit of one here. But this one's almost completely intact. Here's where it's ducted to the surface. You got a hair in this hair follicle, so you know it's emptying into that hair follicle like a sebaceous gland likes to do. We don't completely understand these glands. The upper about 20% of these glands looks like a typical sebaceous gland that's emptying into this hair follicle. The rest of it is atypical. We don't think it even is releasing any secretory product into the hair follicle. Here's a little bit of the hair follicle right here. Look at these cells. They, some people thought that they looked almost like hepatocytes with canaliculi around them. So they called them hepatoid in appearance. Again, we don't know what they do except for they love to become a carcinoma, an adenocarcinoma, adeno referring to gland. And these are the circumanal glands. And as the name implies, they encircle the anus. So they go all the way around 360 degrees around that mucocutaneous margin of the anus. So the circumanal glands, a common site of adenocarcinoma in intact canines. Again, the anal sac with the glands of the anal sac. And then one last gland is the anal glands. And I don't see it listed on here, but it would be in your lab notes. These are just modified sweat glands that secrete an oily product. You know how you know how the mucous membrane, the non-haired skin of the anus, you know, tends to be rather you know, oily, kind of glistens on your dogs. Reminds me of a cartoon. Yeah, anyway. So these are anal glands, and they are modified sweat glands that produce an oily secretory product, anal glands. So how do you know, tackle all these different glands here in this section? Well, it's pretty easy. You've got the anal sac here. So any gland associated with it is the gland of the anal sac. And these are modified sweat glands. We don't have a cat section, so you don't have to look for sebaceous glands down here. And that anal sac ducts out. Here's a little part of it's duct right here, ducts out to the surface right here. This is just normal haired skin. So you'll have sebaceous glands and sweat glands. And then over here, you got your funky little modified sebaceous glands, your circumanal glands that look like your little bits of Christmas stockings here. And how you tell that they're not usual is that they look different. They got all these like canaliculi looking spaces around the individual cells, and they're not as foamy. They're not as filled with lipids as you would expect in a sebaceous gland. And then that just leaves over here near the, or surrounding that anal sphincter where you have both smooth and skeletal muscle in here. Then that's just the anal glands, and they secrete that oily product that kind of keeps that skin surface lubricated. Okay, I think the only slide we didn't look at was just the teat, and really that's just more practice. This is closer to the body. This is further away from the body. Just a mare teat, and it's a way to practice more glands. You know how horses like to do things a little differently? So their sweat glands are just massive because you know horses sweat a lot. So look at these massive piles of simple tubular glands that are all just coiled up and in a tight cluster. Here, we've got some smooth muscle fibers here. I think those might be some of the erector pili muscles. And here you've got sebaceous glands. You've got some fine hair follicles because the mare teat does have really fine, small hairs. So here we got little follicles invaginating in. You can see the sebaceous glands crowding around that hair follicle because they're lazy. They just want to secrete into the hair follicle lumen instead of having to make their own duct to empty out onto that skin surface. You can practice your dermal papillae and your epidermal pegs, your papillary layer and your reticular layer. You can practice sebaceous gland ID. You can be amazed at these massive piles of sweat glands so that horse can do what it does. It sweats a lot. And you can practice all the layers of your, or as many layers as you can ID in your stratified squamous epithelium. And this teat had heavily pigmented skin. So you can see that. You can see a lot of melanin. That's it for the class notes, lecture notes up to mammary gland and the digital organs, which would be the equine hoof that we're going to go into. We'll save that for the next lab. Okay. All right. Whew. Integumentary system. It's not that hard, but there's just a lot to it. Okay, Dr. K, you out there?
I'm out there.
All righty.